All right, you guys should have an idea of, oh, look at that. Dude, why is no one in this class all the time? Following me around. All right, I got the whole board now. All right, this is the class next door. If you show up one day and you don't find me in the regular class, just come next door. I like this one better. I don't know why. Yeah, 312? 312, yeah. All right, do you guys have any questions, including a question about like what I might ask? Like, I think I covered everything that I'm going to ask from those two chapters. I talked about the cell, how you have to know the organelles. You need to know the difference between a eukaryote and a prokaryote, and the key word there is bacteria. And then we talked about cell membrane, and I already mentioned those things. So you have a really good idea of half the test now of what I'm going to ask you. So, and hopefully you knew a lot of that already. So, do you all have any questions on any of that?
Are we gonna need to know like the prophase, metaphase, anaphase?
Yeah. So that's what I'm going to do now. I'm going to get into that prophase, metaphase, anaphase thing. But I'm going to tell you now. It's in your notes. Yeah. So let's revisit cell cycle. This is awesome. Okay. So, 3 parts. We can break the cell cycle down into 3 parts. Part 1, interphase. The only thing that I asked you from this, which I might ask again, I don't know how, but like, what part of interphase do we synthesize DNA? Or in what part do we make DNA? Or what part, this question could be asked in a lot of different ways. Where is the genetic material duplicated? I don't know. I mean, it's essentially all the stuff. When are the chromosomes doubled? When are the chromosomes made? That's just another way of saying DNA. Right, and that happens in the S phase. So there were three phases to this. There was G1, there was S, and then there was G2. I didn't really give you any information about G1 or G2, but the only thing you need to know about G1 and G2, they're the same, is all the organelles are doubling. All the ribosomes, all the mitochondria, all the Golgi, they're all doubling. In the S phase, that's where DNA is made. There you go. That's it. That's the first part of the cell cycle. You've got a cell, you got all that shit in there getting doubled up, and that's a big fat cell with a big nucleus and twice as many organelles in it. So it is crowded and ready to split. But first we got to take that one big nucleus and we got to turn it into 2 nuclei. So that's the second phase, mitosis. And we were doing 4 parts to mitosis, but remember some books or some videos are going to have 5. So that's fine too. I was just trying to make it easier. So there was prophase, and in prophase it was chromatin condensed. Before they were not looking like chromosomes. Now they are. And I in a previous lecture I drew it on the board, right? Inside that nucleus, it was just all, here's the nucleus, and it was just all like this. That's what it looked like before. So what happens in prophase? you get what look like all this stuff condenses into what we know as chromosomes, like what we think they look like. And it's different amount, you know. For us, there'd be 23 of these, right? So anyway, that's the difference. This is at the end of interphase. This is prophase, chromatin condensed, and then you have two centrioles, or centrosomes. I don't care which word. Those centrosomes or centrioles that are in there, they start to move apart. They're both here in like red, if I've got red. There they are. And I just want to see if it goes that way too. They're starting to move apart, right? These two centrioles are going to move to opposite ends. So that's prophase. So just two things. If I'm like, what goes on in prophase? That's what goes on in prophase. Metaphase. Chromosomes line up along the metaphase plate. It's an imaginary line. So in other words, it's going to look something like this. I'll write the centrioles. So the centrioles are on opposite ends now. And the chromosomes are all like they're lined up. There's like an imaginary line right here. That'd be like the metaphase plate. So they're all lined up. That was the second phase. The third phase, anaphase. So I guess I only got one thing here. You might have two in your notes. If I put something second, fine. You know, if that's all you got, fine. I might have said something about maybe centrioles are at like opposite ends. That's fine. Or if you just want to put that, that's fine too. Anaphase. This is the part that, so if you cannot remember all the words on this, it's okay, but you should be able to write down, you should be able to explain what's happening. So I'm going to just write the words. Kinetochore microtubules retract. And then we have those tubules that are not, well, just write this down and then I'll explain it. So, I don't know if you guys can see the color, but some of these, and I probably made it too complicated in here, but these are called microtubules. Microtubules. I didn't want to write the whole word. They're like those rods that you're sending out. to the nucleus or to the chromosomes. They're called microtubules. Kinetic core and non-kinetic core. If you can't remember that word, just remember what's going on. You've got these tubes that are like hooking on to the chromosomes. Those are called kinetic core microtubules. Then you've got some of these rods or whatever they are. They're called microtubules. You have some of them that are not touching the chromosomes, right? So those are the ones that aren't, and I wrote them in blue, we call those non-kinetic core microtubules. And we call the ones I wrote in black, they're called kinetic core microtubules. They're touching the chromosomes. That part of the chromosome that they touch is called the kinetic core region. So what you need to know is that some of these rods are touching the chromosomes and they're going to start pulling back. So they're going to pull these apart. So I'm actually going to pull them apart a little bit. So here they are. Oh, I already screwed it up. So the kinetic core microtubules are shorter now. They started to retract, and you see they're pulling the chromosomes away from each other. But the non-kinetic core microtubules are getting longer. And that's actually, by them getting longer, it's changed the shape of the cell. It's more oblong now, because these non-kinetic core microtubules they're actually like pushing on each other and making the cell get longer. So that's what's happening in anaphase. The kinetic core microtubules are retracting. That's pulling the chromosomes, because now you can see you've got like a cluster here, that's going to be a new nucleus. You have another cluster here, and that's going to be another nucleus. That's the next phase, right? So you see how it's going into telophase. As you can see it. In telophase, you have two nuclei. I didn't probably put this on the last one. But I mean, it's okay whether if you include it or don't include it, but in telophase, you have one, and I'm gonna write this new one here. You've got one, cell, and you got like, here's the new nucleus, it's not quite formed, but there it is. And you got two nuclei. And in fact, you can even see right here where the cell is starting to split. You can see how it's going to break apart. So that's telophase. Nuclear envelope, that's the word I have here. That's the envelope is surrounding the nucleus. The nucleus has a membrane around it. That's called the nuclear envelope. What happened way back here in prophase is that nuclear envelope, it started to break up, but all the pieces, the cell saved it. And then when the new nuclei is made, the two nucleus, when they're made, the nuclear envelope, the pieces that were broken up from the old one is going to be used to go around this. So nuclear envelope is just a membrane for a nucleus. It envelops it, it surrounds it. That's all mitosis. Some books have prometaphase, I skipped it. So two things here, one thing here, two things here, one thing here. That's going to answer any question. I might say, explain the phases of mitosis. I might just take one question, but I'm lazy. I'll probably just say, describe mitosis? Because that's really easy for me. I just write 2 words and that's my question. It's not really easy because I gotta grade it. I mean, you gotta do the work on one end or the other. So that's the second phase of the cell cycle, mitosis. The last phase, that I gotta erase this. That was the hardest part, obviously. And then the third phase is cytokinesis. And I don't got anything to say about that. Just cell. One cell becomes 2 cells. Done. I don't know if that matters, cleavage hurl, but I mean, if you want to include it, you can. But 3 parts here. Interphase, that's like 90% of the cell cycle. Mitosis. Mitosis is when one nucleus becomes 2. Cytokinesis is when one cell becomes 2. Some books are going to include cytokinesis and telophase, they put them together. There's like no clear distinction between number 2 and number 3. Whatever, that's also fine. I don't really care how you put it. But that's what happens. So this cell cycle is a eukaryote, not a prokaryote. If you remember the difference, because I might ask you that on the test, the difference between the 2 types. Prokaryote, bacteria. Eukaryote, everything else. So this is everything else. But this is not bacteria. Bacteria don't go through this. And I think I asked that on the test, but I can't remember. But what do bacteria go through? I forgot how to spell fission. I can't remember how to spell it. They go through binary fission. And it's a really simple process. We're not going to bother with it. Just know that they go through a different process. If you take microbiology, you're going to spend most of your semester on prokaryotes. You're going to learn all about binary fission. It's actually amazing. I'm not giving it the time it deserves. But, you know, we got to move on. So, cell cycle. Know the 3 stages of the cell cycle. Know what happens in each stage. So what happens, if I ask you what happens in interphase, all the organelles double, and then the S phase is the DNA doubling, or replicating, whatever word you want to use. I don't care if it's the word doubling, replicating, making more. There's a shitload of stuff in that cell. That all answers the question. You're not confined by specific words, so don't get hung up. Just know what it does. What happens with the nucleus? You got those, I mean, with mitosis, you got those 4 stages. And then, you know, I put a couple things down for each stage. So know what happens in that stage. Know what cytokinesis is. That's really easy. Cell splitting. I might ask you about this. How is bacteria division different from eukaryotes? Or I might say, how are prokaryotes different from eukaryotes? And you just say, they use binary fission. We have to do the whole cell cycle. And then I would ask you about cancer, which I think a lot of you got right. Those questions, but cancer is when you have the cell cycle without any control, right? It's got to stop. You got to stop dividing cells. You can't keep dividing. So your body has ways to control the division. So That's cancer. And then no stopping it. No, for example, no density dependency. And then this word tells you what it is. It's dependent on how dense it is. The more dense it is, the less likely you want more of something in there. Right? We operate on density dependency. If you get too many people in a room, you don't want to go in that room. If you get to the drive-through and there's 30 cars in it, unless you're at Chick-fil-A, you're like, nah, I'll wait. Too many. Cancer, no. Cancer's going to jump in there anyway. They're not even going to get in the line. They're going to like pull their car up on the side. You're going to have all these cars trying to edge in. That's what cancer is. Just no control. Right? You can't get all those people through the drive-through anyway. So what happens? The parking lot gets too filled. That's cancer. So the key difference in cancer was that is it benign? Does it enter the bloodstream or not? If it enters the bloodstream or the lymph, we call that metastasis. And blood and lymph are the two main ways to get around the body. Once you put something in the lymphatic system or the blood, it can go anywhere. It metastasizes or it goes through metastasis. So that is called, I guess we call that malignant. But if it's not metastasizing, then I think we call that benign, and you can just take it out. So you need to know that. You know what metastasis is. You need to know density dependency. Why is this fitting in here in the cell cycle? Because all the chemotherapy agents, if you're going into nursing or something like that, you're going to take pharmacology. So all this mitosis stuff comes back. Metaphase, anaphase, telophase. You'll see it again. You'll have to remember all of that again when you're talking about chemotherapy agents. All right. That is everything from the cell cycle. I think you got all the questions. There's probably like 3 or 4 questions from that. This last one, cell respiration, I'm going to really try to narrow it down to the things I'm going to ask you about in particular. So, my first question that I would ask you is, what is the point of cell respiration? Because if you don't know what the point is, then all the details are useless. You're going to forget them because none of it matters. The purpose of cell respiration is to make ATP, take our food and make ATP. Take energy from food and make ATP. That's it. We're transferring energy. Oh, I get it. When I move, it takes it off what I just wrote. Take all the energy from our food, and that energy in our food is going to be in the form of electrons. We're taking the electrons off our food, and we're using those electrons to put together ADP and P. I'm going to even write that because I like that. Remember, the parts of The two parts of ATP, ADP, the adenosine diphosphate, and then the inorganic phosphate, the P with sometimes it's got that little I written next to it. We want to put these together. Whenever you put two things together, you need energy. It's an anabolic process, right? So ultimately, it's going to be through the proton gradient, but really, it's coming from the electrons. Take the electrons off food, use the electrons to put together the ADP and the P. So there's actually like 3 steps to this. There, I've actually given you the main idea of the 3 stages of this. This is glycolysis, the first one. Get that sugar ready, because we're going to rip all the electrons off, but you got to prepare it. Can't just leave it as glucose. You got to do some stuff to it first. Then it goes into the Krebs cycle. And what happens in the Krebs cycle? You're taking the electrons, harvesting electrons. That's essentially what it's doing. Then that third stage, electron transport chain. You're taking those electrons and you're doing some stuff with it. You're turning on these pumps and the H pluses and all that. But ultimately, you're going to make ATP from it. So these are the 3 stages: glycolysis, Krebs cycle, electron transport chain. It's just a good idea if you could know that in your head. When you look at it that way, it doesn't seem too hard. Get the sugar ready. Take the electrons off of it. Use those electrons to make ATP. But instead, I'm going to put the words, the 3 stages. Stage 1, glycolysis. Ultimately, we're getting 2 things out of this. One, and most important, we're getting pyruvate. So we're essentially taking a molecule that's 6 carbons long and we're breaking it in half into 2 3-carbon molecules. That's basically what glycolysis does. It's a long process, but at the end of it, you end up with 2 half sugars. 2 half glucose molecules. So going in, a molecule of glucose. Coming out, 2 half glucoses. Pyruvate. 2 pyruvate, and we get some ATP from it because you're breaking up something, right? So you get some energy. Whenever you break up something, you get energy. So we have to spend some energy to get energy. So we spend some ATP in this process, but we got more ATP out of it. So when I put net, I mean profit. That's not a big deal though. 2 ATP, big deal. It's not a lot. Now, before we go into the Krebs cycle, so this is stage 1. I'm going to do a stage 1.5. We got the pyruvate is too large. We have to make it smaller. So in this stage, we take pyruvate, we convert it to that acetyl coenzyme A. How do we do it? We take pyruvate, step 1, rip off CO2. Step 2, add coenzyme A. And that's it. If you don't remember that, I don't really even care. I'm just trying to let you see what it is we're doing different. Rip off a carbon dioxide, so pyruvate is 3 carbons, acetyl-CoA 2 carbons. That's the whole thing about step 1.5. 3 carbons is still too big. It's big enough to get into the cell, like 3 carbons is fine. The cell can take it in, but we gotta get it into the mitochondria. 3 carbons is too big of a molecule. It won't fit into the mitochondria. So we gotta make it 2 carbons. So rip off one of the carbons. We're gonna add oxygen to it and take it out, breathe it out. So rip off one of the carbons, add this thing called coenzyme A. There you have the word. That's what CoA stands for. So then we call it acetyl-CoA. This is what's going to enter the Krebs cycle. So if I ask you what enters the Krebs cycle, don't say pyruvate. Say acetyl-CoA. So we're tweaking it again. We're getting pyruvate ready to be ripped apart. We're preparing it. So we got to tweak it a little bit. Yeah, I'm going to erase step 1.5 because I want to put step 2 in here instead. Step 2, Krebs cycle or citric acid cycle. You probably you learned citric acid cycle. They've been using that word for a long time. I like shorter words. So what are we getting out of the Krebs cycle? If you tell me in one word, like what's the purpose of the Krebs cycle, just give me one word, what would that word be? At the end of all of this, what are we trying to do? And I'm writing 2 words. The one word would be electrons. We want to get the electrons. By the way, this Krebs cycle, this glycolysis happens in, can you see that? This happens in the cytosol. I don't know if you can even read that word. Maybe you can. This happens in the cytosol, step 1, glycolysis. The other 2 steps, I hope you're saying mitochondria. Long words, I don't know how you guys memorize all these words. Krebs cycle happens in the mitochondria. Get the electrons off of the acetyl-CoA. Rip the acetyl-CoA up. It's nothing now. It's just atoms. That's it. Nothing. Your food's gone now. Get the electrons off of it. We get some ATP. We get 2 ATP out of this. Not a big deal. The big deal is getting those electrons. And when you look at the Krebs cycle, you're going to see a lot of these molecules called, if you remember, FAD. They're going to start with FAD or NAD. FADPH, NADPH, NAD minus plus, they're getting oxidized, they're getting reduced. We never talked about any of that. So I just told you no FAD, no NAD. They're just molecules that grab electrons from one place, carry them to another place. If you study nutrition, these are all B vitamins. Like this whole process, you've got like 8 B vitamins or 9, something like that. Around 6 of them are being used for this whole process. Coenzyme A, that's one of your B vitamins. That's a B vitamin. That's another B vitamin. There's lots of B vitamins here. That's different. So this is the Krebs cycle. This is what we're getting. We're getting electrons, we're getting 2 ATP. These are just electron carriers. I'm gonna even erase it. So I hate when you guys write that on the test. You'll be like, oh, we get 2 ATP NADPH. You think it's like one word because you wrote it in your notes. And then you studied it just like you wrote it. And I get that. I do the same thing. This is what you're getting. Just go back to the purpose of the whole thing. We want to get the electrons off the food. There we go, done. Now we're going to use those electrons, step 3. So I'm going to erase all this and put step 3. So far, before I go to step 3, what would I ask you about? I'd ask you, what are we getting out of glycolysis? That's a question, right there. I could just switch the word. What are we getting out of a race glycolysis? I'll put Krebs cycle. That's another question I could ask. That is the type of question I will ask. I'm going to ask one of them. I don't know which one. Or I might ask, like, what goes into the Krebs cycle? Like, what's the molecule that goes into the Krebs cycle? Where does the Krebs cycle happen? Where does it occur? Right? Mitochondria. So those are questions. One other question. What is the purpose of cellular respiration? What's the whole point of all this? That was what I wrote first, the first thing that I wrote. You should know that. Because that answers the question, why are we breathing? Like, why are we breathing? We do it all the time. At least most of you do. Why are you doing it? For all this. We need to get the oxygen in there because you keep breaking down food, and we got to get rid of all that carbon and stuff. So that's why we're breathing. So anyway, step 3, electron transport chain. Electron transport chain. You will write out on the test somewhere, electron transport chain. I hate when you memorize ETC and you have no idea what it is, and you have no idea what it does. If you copy down exactly what I put on the board, I hate that because it just shows you're good at memorizing random words that you're going to forget. Right? I know you're going to forget it. Why would you remember ETC? That doesn't mean anything. It's just 3 letters. I know, why would you remember electron transport chain? I get it, but I want you to explain what's going on with these things. We're going to take the electrons and we're going to use it ultimately to make ATP. So, take the electrons. Now I'm going to put that because I'm too lazy to write electrons. Take the electrons, turn on pumps. Not just any kind of pumps. Turn on what pumps? I hope you wrote that. Those H+ pumps, the proton pumps. We have this phospholipid bilayer. And then we have these pumps. Wish I had a better black pen. All right. This in blue is supposed to be the membrane that's inside the mitochondria. It's a phospholipid bilayer. We've got protons, that's what an H+ is, because when you take the electron off of a hydrogen, it's only a proton. That's all that's left. So we just call these protons. Your body uses them all the time. everywhere. You're gonna see protons in lots of different things. Your body uses them to do a lot of stuff. Here, we're gonna use them to make ATP. So we're gonna take the electrons from our food that we got in the Krebs cycle, turn these pumps on. Not this is not a pump. We're gonna turn these pumps on, and we're gonna put all the protons on one side. So the electrons come through here, turn these 3 pumps on. Now I'm gonna put my H pluses on this side. Here's all my protons now. But that's active transport, right? There were 5 here, and there were 5 here. I took the 5 from here and I put them against the fusion. Against the concentration gradient, I put them here. So I needed energy to do that. That was these pumps. That was the electrons. With the electrons, I turned these pumps on and I put them all on one side. I've created, what have I created here? A proton gradient. Oh, God damn. Tell us not to close the doors because of the coronavirus. So yeah, let it all. Are you okay with me not wearing a mask? You okay with me not?
Yeah.
Okay.
That's good.
If we weren't this far apart, I would definitely wear one for you. And if there were more students in here, I'd wear one. I'd put it on for sure. That's a proton gradient. They're all on one side, and there's none on the other side. These want to diffuse, right? That's diffusion. Higher concentration to lower concentration. These want to go to here until they're equal. They're going to come right through here. We're going to let them through, but only through here. Come on through. Bring them through there. That's this machine. It's not really a machine. We're going to pretend it's a machine. This machine, the ATP 3000, used to be called ATP 2000 20 years ago. The ATP 3000 puts, what are the 2 components of ATP? Puts them together. So sitting inside here, inside this machine, you got a bunch of ADP. You have a bunch of P's. They're all sitting in this machine. You just got to put them together. So when all of these protons are coming through, it's like water turning that wheel. They're all coming through. That's putting the P and the ADP together. That's putting these 2 together. And we get we get 30, something like 34 ATP from this. So there's 2 different types of energy things going on here. Number 1, the electrons turn the pumps on to make this gradient. That's an energy. This is energy. When you have all the H pluses on one side, that's stored up energy because they want to flow through here now. So the proton gradient is what is putting the ATP, the ADP, and the P together. So if I ask you, what is immediately creating, I think that's what I put on the test if I did that question. What is the immediate energy that's creating ATP? It's the proton gradient. How did we get this proton gradient in the first place? The electrons. The electrons turn the pumps on, and that allowed us to shove all the H pluses on one side, all the protons on one side. The electrons allowed us to do this. Now when they start flowing through, that's making the ATP. That's the proton gradient. The question is, how do you explain it on a test? My answer is in your own words. Pretend like you've watched this a few times. You know, we rewind 5 minutes, watch it like 2 or 3 times, and then you go tell your mom. Just whatever you can remember in your own words. That's what I want. And then you just write that on paper in your words. Just however you're talking to your mom. Hey mom, you get these electrons from food and they turn on these pumps to put all those H pluses on one side. They're coming through one place and that's like a watermill. It puts these two together. Like you could put the ADP and the P together. If you can say something like that, that's great. You didn't use the word proton gradient. I don't care. You understand what's going on. I don't care if you don't remember 34. You go out to your car, you're like, oh shit, I put 35. I put 30. So what? It's still correct. I mean, it's not correct, but whatever. You understand what's going on. And you miss some little stupid number. Is anyone ever gonna? Yeah, on the test to get into nursing school, they ask this stupid question. How many ATP are made? By the way, it's 38 because you got to add 2 from glycolysis, and you got to add 2 from Krebs cycle. So you add all of them, but anyway. In nursing school, you're going to have to know 30, 40, no. I'm never going to expect you to know that. You have to understand this, yes. So, how does the electron transport chain work? That's a question. Or maybe I can find some questions within this. What's the immediate source of energy that creates ATP? And you know where this happens, right? This thing that I was writing here, this is all the mitochondria. This is all inside the mitochondria. Because remember, that's what mitochondria does. Think back to the very beginning of class. I put mitochondria, and then after that I put makes ATP. Right? This is the organelle that makes ATP. You've got all the questions. Maybe I'll put like 10 questions on the test. You've got all the questions. You know what I'm going to ask you. You know what I'm expecting for an answer. It's like, if you understand it, and you just write in a way that shows me you understand, then that's good. I would rather, it's better for you to understand it first, then start trying to memorize words. If you try to reverse that, and just try to memorize words, you're not going to do it. You're not going to make any sense. You're just going to be writing words, and you can't just throw down words. It's not acceptable. Rather you understand and miss words than write a bunch of words without understanding. You didn't put proton, you didn't put a particular word. It's okay. You spelled phagocytosis wrong, big deal. You know what it is? You just forgot? You wrote it with an F. F-A-G, racist, but I'm not going to mark you down for that. Even though I guess I should. All right, you guys have any questions? You know what it's going to be. All you got to do is just put in the time. Don't wait until tomorrow night. That is what F's you up on every test for every class. Keep waiting till the last minute, some of you. Not all of you. Don't do that. Today, get it all in your head. Lock this whole thing in by tonight. You can explain all of this tonight. Then go to bed. You're going to wake up. You're going to forget some of it. It's normal. Do it again. Go to bed tomorrow night. Wake up Wednesday. You go open your notes, you don't need it, you know it. You're like, I don't know what to read, I know all this, I know, and then you really know it. And then come take your test, you get 100, and we're gonna turn all this around, I'm gonna take your F and I'm gonna put an A, your grade's gonna go up, and you've done the discussions that you haven't done, then your grade's gonna go up again, and then you're gonna see that all this is gonna be okay. And I'm gonna feel better, and not get so depressed. It's gonna be a great class.
What time and what room are we doing the exam in?
Oh, yeah, good question. Let me see, hold on. Not this one's over here. Yeah. No. Okay. Yeah. Okay. Great. When's 312? 312, right next door to the regular room. The times, we're going to use the same times that I sent you in that previous announcement. So look up the previous announcement, depends on what your last name starts with. If it starts with an A, for example, you're at 8:30. If it starts with X, you're at the 11:30 slot. Right,
Okay,
so
this might be a little ridiculous, but I haven't been to campus yet. What building?
the main 3-story
Okay.
building.
Thank
It's
you.
on the 3rd floor. That's good, we don't want you on campus. Give us coronavirus. All right, do you have any other questions? All right, please study. I'm gonna post these two today, in case you miss anything. I think you have everything from today. It'll be in 2 lectures, right? Because we broke for grade. So I'll put them up as 2 lectures. So look them over if you have any other questions. Look at other videos. There's much better videos. Do Crash Course, Amoeba Sisters, Khan Academy, whatever you want. And that's it. I'm gonna see you guys on Wednesday.